Bismillahirrahmanirrahim. Assalamu alaikum. My name is Zaryab from BS Physics. The topic that I'm going to discuss that one is a steroid. Let's see what is steroid. Steroid possess a common structure per hydrocyclopentanophenoethylene nucleus includes cholesterol, bile acids, androgens, adrenocortical, adrenomedullary, estrogenic, and progestational hormones. We can see that the structures of the steroid containing 4 rings of the carbon atom, 3 6 membered and 1 is 5. Actually, steroid includes many hormones, alkaloids and vitamins. Next, I will discuss the classification of the steroid. In the classification of the steroid, first one is a sterol. Sterol, also known as a steroid alcohols, occur naturally in plants, animals, fungi, with the most familiar type of the animal sterols being cholesterol. Now I will discuss the types of the sterols. Phytosterols, plant sterols, campestrols, cytosterols, and stigmasterols. Blocks cholesterol's absorption site in the human intestines, thus helping to reduce the cholesterol in humans. Zoosterols, animal sterols, cholesterol. Ergosterols, sterol present in the cell membrane of the fungi. Actually, sterols are a class of the hydrophobic ringed lipids. Molecule found in the biological membranes throughout eukarya. Their actions are thought to be essential to the formation's lipid rafts with a various effect on the biology and serve as a precursors to steroid hormones in animals. Next one is a bile acid. Bile acid are steroid acids found predominantly in the bile of the mammals and other vertebrates, are conjugated with the taurines or glycines in the liver forming bile salts. Actually, there are two bile acids. First one is a primary bile acid and second is secondary bile acid. Primary bile acids are those synthesized by liver. Secondary bile acid result from a bacterial actions in the colon. Now I will discuss the two functions of the bile acid. First one is to remove unwanted cholesterol from the body and second one is that to aid in lipid digestions in the intestine. Bile acids comprise about 80% of the organic compounds in the bile. Other are phospholipids and cholesterol. Next one is the cardiac glycosides. Cardiac glycosides are glycosides of the mostly carbon 23 steroidal compounds are called cardiac glycosides because they modify heart actions. If we see the medicinal importance, they are cardiotonics used to treat congestive heart failure, CHF. They increase the force of the contraction of the cardiac muscles without increased oxygen consumption, increase cardiac output. The diastolic phase lasts longer so decrease heart rate, have diuretic effect due to increased amount of the blood passing through the kidney. Actually, cardiac glycosides are a class of the organic compound that increase the output force of the heart and increase its rate of the contraction by acting on the cellular sodium potassium ATPase pump. Next one is the steroid hormones. A steroid that acts as a hormone can be grouped into 5 groups by the receptor to which they bind: glucocorticoids, mineral corticoids, androgens, estrogens, and progesterogens. Steroid hormones help control metabolisms, inflammations, immune functions, salt and water balance, development of sexual characteristics, and the ability to withstand illness and injury. Next, I will discuss the classes of the steroid hormones. Actually, there are 10 classes of the steroid hormones. First one is the glucocorticoids. Glucocorticoids originate the adrenal cortex and affect mainly metabolisms in diverse ways, decrease inflammations and increase resistance to stress. Cortisols. Cortisols dominate the glucocorticoids in humans synthesized from progesterones in the zona fasciculata of adrenal cortex involved in stress adaptations, elevate blood pressures, and sodium positive uptake. Numerous effect on the human system. Mineral corticoids. Mineral corticoids originate in the adrenal cortex and maintain salt and water balance. Aldosterons. Aldosterone, the principal mineral corticoids produced from progesterones in the zona glomerulosa of the adrenal cortex, raises blood pressures and fluid volume, increase sodium positive uptake. Next one is the estrogens. Estrogen originate the adrenal cortex and gonads, primarily affect the maturations and the functions of the secondary sex organs, female sexuals. The natural estrogenic hormones are estradiol, estrogens, estrols produced in the gonad, adrenal, and placenta. Estradiol and estrogens, principal female sex hormones produced in the ovary, responsible for secondary female sex characteristics. Androgens. Androgens originate in the adrenal cortex and gonads and primarily affect the maturations and the function of the secondary sex organs, male sexual determinations. Testosterone and androgens, male sex hormones synthesized in the test responsible for secondary male sex characteristics, produced from the progesterone's most potent androgen in the blood. Testosterone hormone, any of the group of the hormones that belongs to a class of chemical compound known as a steroid. They are secreted by three steroid glands: the adrenal cortex, testes, and ovaries, and during pregnancy by the placenta. All the steroid hormones are derived from cholesterol. Next one is a progestins. Progestins originate from both ovaries and placenta and mediate menstrual cycle and maintain pregnancy. Progesterones. A progestagens produced directly from pregnenolones and secreted from corpus luteum, responsible for changes associated with luteum phase of the menstrual cycle, differentiation factor for mammary glands. Next, I will discuss the identity test for sterols. Identification test name is Rosenheim reactions. In this test, if we add trichloromethanes plus aqueous trichloroacetic acid solutions and then we get red color or blue color, we can say that the compound is sterol. Next identification test is Tortelli-Giaf reactions. In this test, if we add glacial Hag plus solutions of the bromines in trichloromethanes and then we get green color at the interference, we can say that the compound is sterol. Next, identity test for cholesterol. Identification test name is a Lieberman's-Burchard test. In this test, if we add acetic anhydride plus sulfuric acid and then we get green color, we can say that the compound is cholesterol. Next identification test is Salkowski reactions. In this test, if we add trichloromethane solutions plus sulfuric acid and then we get red color, green color fluorescence, we can say that the compound is cholesterol. Next one is the identity test for bile acid. Identification test name is Miley's reactions. In these reactions, if we get blue color in transmitted light and needles which appear yellow in reflected light, we can say that the compound is gallic acid. Next identification test is Hammerstein test. In this test, if we add hydrogen chloride plus alcohol plus heat and then we get violet color, yellow color, we can say that the compound is gallic acid. Next one is Patent-Coffer reaction. In these reactions, if we add reagent plus heat and then we get red color, bluish red in several days, we can say that the compound is gallic acid. Next one is the identity test for cardioglycosides. Identification test is Keller-Kalliani's test. In this test, if we add FeCl3 plus concentrated sulfuric acid, we can say that, and if we get reddish brown color or blue purple, we can say that the compound is deoxy sugars. Next one is Lieberman-Burchard test. In this test, if the reagents used are residues in trichloromethanes plus acetic anhydride plus concentrated sulfuric acid, and we get immediate color change blue, green, red, pink, purple and violet. We can say that the compound is a glycone steroidal group. Next one is identity test for the cardiac glycosides. Identification test is CATS test. And the reagent used are trichloro solutions of residue solutions of the trichloromethanes, CATS reagents A 3,5-dinitrobenzoic acid plus CATS reagents B sodium hydroxide, and then we get violet color. We can say that the compound is unsaturated lactones. Thank you.